All right, greetings and welcome to the very last talk of the JGI user meeting in our technology session. I have the good fortune of introducing Kelly Wrighton, who is an associate professor at Colorado State University in the College of Agricultural Sciences. And Kelly did her graduate and postdoctoral training at UC Berkeley and has since garnered numerous awards and honors, including a DOE Early Career Research Program Award and the Presidential Early Career Award for Scientists and Engineers. The Wrighton Laboratory investigates how microorganisms contribute to ecosystem processes with a particular interest in carbon and nitrogen cycling. And their approach is scaling genomes to ecosystem function in really a very broad range of systems. She's been a JGI collaborator and ambassador since her postdoc days, and she currently serves on the JGI Executive Committee. And a useful piece of trivia, if you've ever listened to JGI's Genome Insider, you have heard her voice as part of the intro sequence to that podcast, and so which I highly recommend. There's one featuring Kelly, but she's on every episode. And I always enjoy hearing her voice on there because I always look forward to her talks because her lab is consistently on the cutting edge of applying novel experimental and informatic techniques to microbiomes. So I'm looking forward to today's talk, which is Window into the Wetland Soil Methane Cycle. And Kelly, I'll let you get going.
Great. Thanks, Susanna. Oh God, I didn't even know that I had that on that something to go obsess about. Okay, so today we're going to talk about the methane cycle. This is going to be largely a new story, so I'm excited to share it with you all. So I don't think we need to talk too much about the importance of methane to this audience, but over a 100-year period, a ton of methane is equivalent in climatic forcing to about 28 tons of CO2, and methane is increasing. Why I think it's such an interesting issue, though, is back to this title slide. It's this idea that there's this microbial world, this invisible microbial world that has these macro scale functions that matter at a global scale. So when we talk about methane in the atmosphere, let's talk about where it's coming from. And what you can see on this figure is that on the orange is the anthropogenic sources, but what I want to draw your attention to is the green arrows. And particularly, I want to draw your attention to the wetlands, so denoted by the white arrow. You can see that wetland soils and the microbial communities metabolizing carbon in those systems contribute up to a third of global methane emissions. And this is on par or normalized to inland waters and oceans and termites, or actually it far exceeds those. Additionally, these systems are thought to be especially climate sensitive. And so this is from the recent IPCC report, but it's basically that there's a large uncertainty in the fluxes from these systems due to warming and other climatic perturbations. And so coastal wetlands, of all the wetland systems, are actually particularly of interest in terms of models and climate change. These systems are basically wetland systems that are housed or impacted by tidal intrusion or tidal impact. And I know all of you in San Francisco, I used to be one of you, does not think of the Midwest as a coast, but others would disagree. So here I'm showing you an image of the continental United States, and you can see those places denoted by coastal wetlands. The field site that I'm going to be talking about today is Old Woman Creek. This is a NER site, so basically it's one of 33 coastal blue carbon sites sponsored by NOAA. And because of that, there's been 20 to 30 years of research science happening on the hydrology and the chemistry and the vegetation, the biology, but not as much on the biology and the fluxes from these sites. And so again, coastal wetlands, what becomes important here is that they're influenced by tidal actions in these large bodies of water. In the case of Lake Erie, what you can see here is this wetland, Old Woman Creek, basically is separated by this levee. And when tidal action in Lake Erie or Lake Erie levels rise, that impacts the hydrology of this system. This is actually happening in real time. And so I started this project out in 2013 when I started at Ohio State as an assistant professor. And what you can see from 2013 to 2018 is the data I'll be talking to you about today, that Lake Erie has actually risen 29 inches in the last 5 years. And so we have this rapid sea or lake level rise in Lake Erie. What that does is it essentially floods over this levee and floods this wetland. And so you can see basically here from this multi-spectral imaging, what's actually happening to the wetland over time. I'm actually showing you in the yellow box on the bottom right, the Old Woman Creek water levels that are at the time of sampling. And then above, you can see this diagram kind of denoting the years. And pay attention to these because they'll keep coming back throughout the talk to kind of orient you of where we are in space and time. So this is a great site for understanding coastal wetlands and the impact of climate on the behavior of methane cycling in these soils. So when we started this project is co-developed by myself and my colleague and friend, Gil Boer, who's a professor at Ohio State. Gil Boer runs the flux portion of our project. And so when we kind of moved and decided we were going to invest resources to build the infrastructure we need to build this Methane Observatory, Gil basically built an eddy covariance tower at the site. It's actually in the Ameriflux network. In addition, we deployed soil pore water dialysis peepers. And this is actually critical because if we want to talk about the methane cycle, we need to talk about it at the resolution that the microbes perform and behave. And so what you're looking at here in this image over here is basically the transects where we've deployed peepers. So every red shape is there's a peeper deployed. And what you see is that we basically have a vegetative, a seasonal mudflat, and an open water site or open channel. And what we're doing is essentially taking and capturing with these dialysis peepers the CO2 and methane and flux that is actually happening, or production that's happening in these soils. Using chamber measurements that are taking measurements of the flux every month, we can then calculate the methane production in situ at centimeter resolve scales. And so this is just to show you that actually Old Woman Creek is a bit of an anomaly. It actually has extreme methane fluxes. And so here you can see the annual methane flux from Old Woman Creek compared to other wetland systems. And down here you can see that the daily magnitude of methane flux is on par with CO2. So this seems like a perfect model system, and I'm going to tell you we got lucky, for studying methanogens and the methane cycle more broadly in coastal wetland systems. So now we're going to talk about the infrastructure on the microbiome side, and this is the part of the project that I lead. And so we've named our site Muk, our infrastructure, our database Muk. And so any of you who've ever sampled in a wetland can probably appreciate this name. This is the stuff you can't get off the bottom of your boots. But Muk also stands for, you probably didn't know this, Multiomics to understand climate change. And I want to stress the fact that this is not a flat genome database. This is a whole suite of the chemical and biological processes in these systems. And I'm going to explain them to you now. But what you can see at every peeper site, when we take a peeper measurement, we also take a surrounding cores. And we always take triplicate cores from a 1 meter position from the peepers. Oftentimes, we will sample at very narrow resolutions in space. We always, though, sample the surface and the deep to look at the differences in the microbial communities functioning at surface and deep. But many times, like in 2018, we sampled at actually 5 centimeter scale resolutions through these soil cores. And so that's some of the data you'll be seeing today. And so the questions we're really excited about asking is, what are the methanogens doing in these systems? And how does it relate to the in situ fluxes or production and fluxes that we're measuring at the site? And so let's talk a little bit more about this Multiomics database. Every time we've gone to the site, anytime we pull a core, we take a 16S and a geochemical measurement from every sample. So for every surface and deep, there's 2 samples. And so over the years that we've been out there, we have over 730 samples, and everyone is cataloged with this baseline data. From that, our goal is to build a methane genome resolved database that underpins a lot of this. And so we really want to sample where the methanogens are, who the methanogens are, how they're changing across gradients, and ask the question of how deeply do we need to sample in metagenomics to exhaustively or get close to sampling the diversity that we see in the archaeal methanogen pool by 16S. And then with those kinds of gradients that we use from the geochemistry in 16S, we can develop questions that we use targeted chemical profiling of the soil metabolome via NMR, FTICR, and LCMS, as well as we overlay metatranscriptomes. And so a resource like this, you can imagine, would not be possible for an assistant professor like myself to build without collaborations. And this really highlights the kind of power and research that's possible through collaborations with EMSL and JGI. So let me tell you a little bit about a methanogen microbial community. It turns out when we look at a wetland microbial community, the most dominant organism in that community is about 1.5%. Everything else, and there's about 1,000 to 6,000 members after that, is decreasing in abundance. So we've got a very even community that has a high richness. Methanogens are really fascinating. They're what we would arguably, you could make the case that they're probably keystone species, meaning their ecosystem effect is disproportionate to their abundance in the community. They sit at that rare portion of that microbial community. So if we want to sample methanogens, we need to sequence through so much of that microbial community. And because of this, this is why these organisms, while they have a huge ecosystem impact, are really poorly sampled in all of our metagenomic investigations. I mean, the fact of the matter is most studies recover 1 to 5 methanogens, and we call that a win. And so you really need deep sequencing at one level to get there. And that was possible through our collaboration with JGI. And so here I'm comparing all the basically library sizes that are in SRA from Wetlands. Now I'm comparing the depth of sequencing that we have at our site. And again, Chris Miller and I, one of my collaborators, we invested a lot of side startup money to also do some really deep sequence to try to get some of the really rare members that we were seeking. From that data, what do we do with all this data? This is not just like a plug and chug assembly to get genomes. In fact, there's 2 dedicated postdocs, and Dr. Penfei Li and Adrian Nero, who actually manned the assemblies of all of these samples over time. And this was an iterative assembly, single cell or single samples, subtractive, sub-assemblies, any different way that we could do it to target those methanogens, we were after. And then we also did differential binning. So again, I want to give you the sense that we use that 16S to highlight the methanogen diversity and the gradients that they operated. We went in because we knew where those methanogens were in the community and did these targeted sequencing of them. And then we brought those genomes to light using a series of targeted investigations. Doing all of this, we recovered over 17,000 metagenome assembled genomes, again, from the whole microbial community. Of those, 370 are methanogens. When we de-replicate those genomes at a 99% identity, we actually recover still 89 unique, high quality and medium quality methanogen genomes. And that is really powerful. But genomes aren't enough. If we really want to talk about putting microbes into models and understanding their physiology, we need more than genomes. And so that's where this suite of chemistry comes in and exometabolome data. And additionally, all these metatranscriptomes come in because the metatranscriptomes let us know what organisms are active and what they're doing, and we compare that to the chemical profile in the system. All right, so let's take a look at that methanogen database. Basically, what we see is that there's 4, and depending on how you classify them, 5 phyla of methanogens that we recovered genomes from. Again, not only is it the number of genomes from methanogens that we're discovering, but compared to any other metagenome study, it's the broad diversity of methanogens. And I really do believe it's because we were targeting them specifically by the samples we chose for our metagenomes. One of these orders, anything in black, is a novel order. So you can see here we have a novel family, and then we have some novel genera as well. A majority of these, like I said, are medium quality, and even many of them are high quality. And what I want to draw your attention to now is, say we're looking at the Methanothrix genus. This is a genus we're going to be talking about a lot in the early part of this talk. You can see we have 16 members from that genus. Many of these are different strains. And so this really allows us to start thinking about questions of niche and how the scale of diversity that matters in these systems. Because we have good genomes, we can also assess the metabolisms of these microorganisms. And that's what I'm denoting here. I also want to draw your attention, this red bar on the top is actually the methanotrophs in the story as well. What I'm now showing you is basically the max transcription of the most active genome in that group, in that lineage. And what I was sort of surprised to see is I thought, okay, we have 89 genomes, but we're going to have this seed bank or this reservoir of methanogens, and maybe 5 or 6 are going to be active. Because that's largely how we think about methanogens from multiomics, because we're just not sampling the depth of them, I think. And what we see is actually this whole range across each representative. There's a very active methanogen at some time point in some space. We'll talk more about that. Okay, so I want to talk about what our knowledge gaps were and what our framing logic was. And so to do that, we should talk about what kind of information, when models assume or ingest or take in information from biology, what are the kind of assumptions that they're making about biology in these systems? And I will say that a lot of this knowledge comes from pure culture work that was done in the 1970s till the mid-90s, growing methanogens largely in pure culture in a liquid broth. And we can imagine that methanogens in the wild, methanogens in a soil matrix, behave very differently than how they would behave in the laboratory. But the first assumption that's widely incorporated into soil microbiology textbooks, and I know this because I teach soil micro, and also implicit in the models, is that methanogens are not active where oxygen is present. In fact, you can think of it as methanogens are only active in deep anoxic soils. And they're actually largely controlled, or the activity of methanogens is really controlled by the dissolved oxygen concentration. The next assumption that's widely incorporated, and this is still even in our microbiome literature, if you go to the literature, if you look at where the methane comes from in wetlands, acetoclastic is the top dog, so to speak, but hydranotrophic follows close behind. And there's very little appreciation for methane in these natural systems from other sources. And so again, most of the methane from wetland systems does not come from methylated compounds. And last, the assumption is that because methanogens are so anaerobic, and this I'm not refuting, they are actually highly sensitive to climatic perturbations. And so anything that drains the wetland and increases the redox would make methanogens sensitive. Anything that floods the wetland would basically make the situation more anoxic and maybe stimulate methanogens. And so understanding how methanogens are tuned to the surrounding environment, assume based on their physiology that they would have a certain performance. And so we're going to go through each one of these and look at our data in light of these assumptions. And so methanogens are only active in deep anoxic soils. This is work that was led by Jordan Engel, a grad student in my lab. In 2014, it started. And so in here, what you can see, this data was actually collected in 2015. And so at this point, the wetland was actually, the barrier breach had broken and the wetland was draining. And so because the wetland had a hydrologic pull, we actually were able to measure dissolved oxygen. And these mud flats were exposed. And so we can see dissolved oxygen in these surface soils. We would expect that the red denotes when the methanogens would be active in the deeps. When we look at the in situ methane from our paired pore water dialysis from those peepers that are located right next to it, we see that the in situ methane is much higher in the surface soils. We then look at the MCRA transcript levels and we see that their methanogens are inferred to be 9 times more active in these surface soils. And so when we look at who is contributing to that MCRA signal, it's anywhere from about 50% to 90% the members of this Methanothrix genus, the one that I pointed out to you earlier. What we also did is we teamed up with Bill Riley and Tim Morin to say, how much does this matter? How much does the activity in the surfaces matter? And so what we're looking at is the data, the methane production data from these peepers, normalized to what we see in the flux. And we can see these methane hotspots in the surface soils where in August, when we measured our meta-T, where these Methanothrix were really active. And during that time, we can infer that this surface methanogenesis contributed upwards of 80% of the wetland methane emissions. And so this is actually called the Methane Paradox. I'm not the only one to study this. And in fact, since our paper came out, a lot of other papers in lake and soil systems and around the same time as our paper even have come out to say that this kind of assumption that methanogenesis is relegated to the most anoxic pockets of soil that you can measure is probably not accurate. From the lab, people were speculating that the methanogens actually had more oxygen tolerance than maybe we ever appreciated. And so what you're looking at here is just a comparison. I I don't think that the methanogens in this system are even seeing the oxygen and dealing with it from this meta-T data. So we basically mined out all the oxygen detoxification genes. If these organisms were actually seeing the oxygen and using this mechanism, you would see it be red and yellow like you see for the above energy generation steps. But that's not what we see. So instead, we propose and support another model in which the methanogens are thriving in these anoxic microsites or biofilms, in which they're probably not even seeing the oxygen. And it's either being consumed by heterotrophs upstream, or that it's actually being protectively omitted from the kind of biofilm enclosure. So refining that hypothesis, I think that there's this great appreciation that measuring the bulk dissolved oxygen concentration of soils is not a good proxy for determining where methanogens are active. The next part of this kind of implicit assumptions that we wanted to look at is this idea of acetate and hydrogen as the main substrates that fuel soil methane flux. I'm not here to actually refute this one because what I just showed you in 2015, those methanothrix, those are strictly acetoclastic methanogens. And I just showed you that in 50 to 90% of the methane flux could be attributed, well, not methane flux, the MCRA activity could be attributed to those methanothrix lineages. However, as we started building this bigger database, what became more clear is that there was a lot of other ways these methanogens were getting substrates. And so, and I wanted to draw your attention to this Welty paper that came out in 2020. It's a really great one for those of you interested in this subject, but basically through metagenomics and through some physiological and biochemical characterizations, there's a whole new world of substrate expansion for methanogens. And it's especially impacted this methyllotrophic lineages. And we're finding these methyllotrophic lineages are much more diverse than we previously thought and use a wider substrate range than we previously thought. And so here I'm showing you the canonical substrates for methanogenesis, because we were interested in how prevalent are these substrates. And oftentimes as microbiologists, we'll go out to the environment and we'll look for the substrates as a cue to what could be important for our bugs to grow on. And we did this. We actually committed this error ourselves. And looking at this data, we assumed that methanol was really probably an important source for the methanogens. When you look at our mags, we have about one mag that looks like it has the methyl transferases for utilizing methanol. And in fact, and that's one's barely or sparsely on in our meta-T data most years. And so in fact, maybe this methanol is in fact the exact opposite. It's actually accumulating because it's not being used as much. It's an interesting idea. So the other point I want to make is when we say that acetate and hydrogen are the dominant substrates for methanogenesis, typically people test a single compound, and they often test methanol as the other alternative. In fact, a lot of our isotopic signatures are not based on any of those other compounds I was just showing you. They're based on the signal from methanol. And so here you can see that we, working with Trent Northern through JGI CSP and looking at the LCMS data, we really want to profile the substrate landscape for these methanogens in these soils. And we're just starting out. I'm just showing you depth profile, but it becomes consistent with our mags. We see that the substrates are abundant and prevalent in this wetland. I will tell you, I also work in permafrost soils, and we're seeing the same thing here. So I really think that we need to give closer attention to the types of substrates these microorganisms may have available to them and recognize that there's been this unappreciated or untapped resource that these methanogens are probably actually taking advantage of. Well, do we have any evidence for that at the genome or the transcriptome level? And so the two phyla that are obligatorily methylotrophic are the crens and these thermoplasmatodas. The crens are also known as verstrati, and I'm going to call them the verstratis because that's how I learned them. And these ones down here are also called the MX, and we're going to go with that because this is a real tongue twister. So when we look at these genomes, because we have such good genomes, we're not really interested in the expression of one gene or two genes. When we talk about organisms or genomes being active in our soil, we have a requirement that 30% of your genome is turned on. And when we look at those genomes that have 30% of their genomes, of their genes expressed, we see that a large portion of those are the methyllotrophic methanogens. The pink ones are also the methanotrophs, I should say. Now what I'm showing you is a genome resolved view of the methanogens over time. So we're looking at 3 depth compartments, depth 1, depth 3, and depth 5. So this is 0 to 5, this is 10 to 15, and this is around 25 centimeters deep. And what you can see is that the methanogens that are using these methylamines, like quaternary amines in this case, or methyl substrates bind to sulfur, are actually some of the most active lineages in these systems. In fact, this Frustrati at many times is actually the most active methanogen in the wetland. And so, like I was harking back to, is, I kind of expected that we would see these substrate gradients. We saw methanol increased in the deeps, we saw more formate in the surfaces, and we'd see the methanogens kind of localized along these substrate gradients. We'd also only see maybe a subset of our genomic pool was active. That is in fact not what we're seeing. So what I'm showing you in this heat map is that we have 89 methanogen genomes, and I'm now tracking the expression of that genome through time on the y-axis. And what you can see is, like, this genome right here is this group of genomes down here is most active, not in the surface soils, but most active in these deep, the middle and deep in July. As we move to August, it's most active in the deep. As we move to September, it's again active in the middle and the deep. And what was so surprising to us is that these were basically organisms with mixed physiologies. So there was not this clear signal or this kind of, we couldn't predict the activity from the chemical landscape. And then you can see that these coordinated groups of transcribing organisms exist. And so when we do a co-expression analysis at the genome level, what we see is that here again is our surface-derived, one of our surface-derived subnetworks. And this green group is our methanothrix. Our Frustrati are down here. These are actually really important and highly predictive of in situ methane. And our methanotrophs are over here. So when we overlay these functional neighborhoods on our methane production, what we can see is that this black group predicts methane production both in 2015 and in 2018. And so we have this kind of dynamic soil community, kind of whittling it down to these functional neighborhoods that are probably really important for methane flux. But despite all of these changes in the microbial community, there is no significant change in the overall methane flux from these sites over time. And so really what that says is the players change, the functions and change with depth and within these groups, but the outputs are really persistent. And so I'd like to leave open the fact that these methyl S and methyl N methanogens are very active in the wetland. They're redundant. I mean, there's tons of them, just like the other lineages, and they're coordinated across wetland gradients. And so I think that we need to do a better job kind of understanding the physiology of these methanogens before we can completely roll out their impact on the methane cycle. And so the last part of my wetlands talk is investigating response to change. And so the question we're asking is, what if a wetland gets wetter? And so here are the nice pictures I took, or I drew for you to kind of highlight what's happening in the wetland. But here are the actual field, the messy field camera shots from the field. And so you can see 2013, we have an unsaturated mud. When Gil and I deployed the Ameriflux tower, we were sampling, this was an island. We were sampling all over this island. In 2015, you can see Garrett, who's a PhD student, who did a lot of the methanotrophy work from our lab. He's taking some cores, and he's, in some, we would say semi-saturated, depending on where you were on this mud flat. But this is definitely a transitional. In 2018, the same site, exact same site, is now under 6 feet of water. And so this was an incredible opportunity to ask what our methanogens had been doing during this time and how they had changed. And we would predict that the methanogens are highly sensitive, and due to a decreasing redox, so more anoxic conditions that happen with flooding, the methane from these systems would be greater, and the methane production in the soils would be higher, and the methanogens would be more enriched. At least that's what that would be my working hypothesis.
Kelly, just to warn you, we are at the end of the time. We don't have another speaker, so it's fine to run over a little. But
well, I'll just say it now. I think I can do it. Well, one slide.
Yeah.
The point of this story is that the methanogens are active and they're stable. We're not seeing them respond to unsaturated or saturated conditions, and the bulk of the transcripts of the methanogens is staying stable in the community, and so are the substrates. And so to summarize, what we've learned is that we need to account for anoxic microsites in methane models, and Tim Moran is actually doing this. He's actually putting the production of methane in ecosystems. We need to have a better updated physiological capabilities to account for the incorporation of other compounds. And lastly, I think it's really interesting on the stability, what parts of this network are stable and what parts of this network are vulnerable. And we obviously need more time series data to understand that. And with that, I can leave my my acknowledgements up, but I do want to give a shout out to one very important person, who's Dr. Adrian Narrow, who led all of this work and has really been the kind of force behind this project. And with that, I'd be happy to take any questions if there's time, or stay on. Thanks, Susanna.
All right, thanks so much, Kelly. That was a great talk, and great to hear that you're working on the, sort of cataloging and modeling these substrates for methanogenesis. I think we do have a couple of questions, so I think we can go ahead and answer those quickly. The first one is from Brett Baker, who wants to know if you've seen any mobile genetic elements associated with the methanogens.
No, Brett, we haven't started looking there, but if you would like to collaborate, Brett, we'd be more than willing to share. And I mean that to the whole community. We have a lot on our plate, and I think the more that we can all delve into it. But we haven't, Brett, that hasn't been an area that we've gone. But we've been reading a lot of your papers to help us identify the methylotrophy in these systems.
All right, and then the other one from an audience member is from Taesak Moon, saying, fascinating talk, thanks. Given your data, what would be our climate future in terms of the methane cycle or microbiome diversity? Any speculation or insight to share? Very interesting.
I mean, I put this all together in the last 4 days, right? I haven't had to think about that. Okay, so I think where we are right now is that we have some pretty targeted hypotheses that I was trying to allude to of where to go. But again, I want to use a caveat on the time scale that we measured. I don't feel comfortable actually saying the behavior of the methanogens to flooding. I mean, this was a 2-year, one time point. And so I think we have a lot more work, but what I'm hoping you've seen from the talk is that we have the observatory built. And so now we can actually start observing at the scales that actually matter and that we need for models. And so it's taken us, 4 or 5 years to build it, but I think we're actually here and we can actually start answering those questions with more certainty.
All right, thanks.